The first two seminars in this series address aspects of how the nervous system is put together. In the third talk, we begin to explore how the nervous system works, or how the brain works. Our ability to communicate with each other and with our environment is defined by our sensory organs, which are responsible for our senses: vision, audition, smell, taste, as well as, and touch, as well as our sense of position relative to our environment and the relative positions of our various appendages. Detection of external signals is actually a very primitive ability. Even bacteria can sense chemicals and respond to them and communicate with them. More complex organisms have evolved complex ways to respond to different types of signals, such as light and sound. We understand a great deal about how cells have developed special mechanisms for detecting and responding to signals of different kinds, but there are still a number of mysteries to be explored. One such mystery, how our sense of taste works or how it functions, has been greatly resolved by the work of our speaker today and his colleagues. A native of Chile, Charles Zuker joined the faculty of UC San Diego in 1986 after receiving a PhD at MIT and a postdoctoral stint at Berkeley. Charles is internationally renowned for his research, for which he has received many honors, including membership in the National Academy of Sciences this past year. Having made major contributions to our understanding of mechanisms of vision, Charles and his group have in recent years focused on the chemical senses, particularly on taste, where their work has set the standards of understanding that we enjoy today. It is my pleasure to introduce to you Charles Zuker, who will present a talk on perception, taste, smell, and vision. Charles.
What I'm going to do today is a bit different than the title of the talk. I am not going to tell you how we ultimately perceive input from any of the senses, but instead I'm going to tell you how we scientists use genetics and model systems as a strategy to unravel fundamental biological processes, both in normal and disease states, and use the senses as the platform to describe the strategies. All right? The sight of a Van Gogh, the sound of Mozart, the aroma of a wonderful wine, let's say a Petrus 1961, the touch of a beautiful woman, and of course, the taste of foie gras. These are the 5 basic senses. And their role is to detect physical and chemical stimuli and convert it into a language that the brain can now understand to get a representation of the outside world. Isn't this disturbing, by the way?
Charles, you consider balance another sense?
Yeah, we are. I put it under Meccano reception. So the truth is that we can call Meccano sensation, which we're going to talk about actually, a larger umbrella of senses that will include our sense of hearing, our sense of touch, our sense of balance, and proprioception. And we'll get to that in this second third of the talk. So the role then of the senses at the periphery, our tongue, our eyes, our ears, our nose, is to detect these cues and convert them through a set of complex biochemical reactions into the language of the brain, which is electrical responses. And the brain then, through a set of very complex and extraordinary integration strategies, can get a percept, which is our own perception of the world. And each of our percepts is slightly different from each other. And that's because our own brain will integrate that information slightly differently, depending not only in the way we are individually wired, but also the way that we have learned to interpret such percept. Now, we are not going to talk at all as to what happens in the brain, but we're going to focus on what happens at the periphery: the tongue, the nose, the eyes, and the mechanical senses. Now, the cells responsible for detecting these physical and chemical cues at the periphery are neuron or neuron-like cells, which are no different than the ones that we have in the rest of our nervous system, and are often responsible for responding to either neurotransmitters, or hormones, or other chemical cues. The one difference that sensory cells have, however, is the fact that they respond to different sets of cues. For example, light, tastings, and odorants. And the final output, once again, is an electrical signal. It is the nature of these molecules in here and their interaction with specific receptors on the surface of neurons, and the nature of the biochemical response that gets translated into an electrical one, which has been the focus of our laboratory for the last 15 years. And our work has used 3 different sensory modalities: phototransduction, what happens in your retina; Meccano transduction, what happens in our sense of hearing and balance; and taste transduction, what happens in our tongue. I'm going to start by focusing on photoreceptor function, phototransduction. Now, our strategy to dissect the function of these processes has been to use model organisms which are amenable to genetic manipulation, so that we can begin to uncover the underlying pathways and signaling processes in a way that we can screw around with the animal, which would not be otherwise possible either ethically or biologically with, let's say, humans. And I'm going to show you the kinds of things we could do with these model systems, and we use fruit flies and mice that will give you a feel and a flavor as to how manipulating these models can really get at the heart of what happens in normal and disease states. Now, it is critically important that you choose a system that ultimately be related or similar enough to humans if you want to be able to translate your findings into human function. And after 17 years of work, I can tell you with complete certainty that we, in essence, are nothing more than a big fly. And may I add that in some cases, more than others. Now, fruit flies, as you'll see in a second, have some really fundamental advantages as a model organism. But before I get to that, let me tell you a little bit about what happens in photoreceptor neurons, which ultimately translate a photon of light into an electrical response. Now, in the mammalian eye, the fundamental cell responsible for catching a photon of light is known as a photoreceptor cell. And these are found in the retina in your eye, and they come in 2 different flavors. There is the rod cells, shown here in orange, which are responsible for dim light vision, so they are the ones which are critically important at night or when you're in a movie theater, and the cones, which are responsible for color vision and day vision. Now, these photoreceptor cells, these modified neurons, have a highly specialized compartment, shown here as this little area full of these stacks, which contain the light receptor molecule rhodopsin and the transduction machinery responsible for catching the photon of light and converting that into an electrical response. So this is a diagram again of the photoreceptor neuron. This is that compartment, and this is a blow-up of around 1 billion molecules of rhodopsin found in each individual photoreceptor cell. And the reason the photoreceptor neuron wants to have so many light receptor molecules is because your ability to see very dim light is directly related to your ability to catch that flying photon. And the more receptor molecules you have, the more likely you are to catch such a photon. Because if the first one misses it, then you have a second and a third and a fourth and a fifth and so forth. Now, the conversion of the photoreceptor signal by rhodopsin into an electrical one involves a very complex biochemical cascade that ultimately translates a change in the conformation of the rhodopsin molecule. So rhodopsin is sitting like this. It catches a photon of light, and it undergoes a conformational change, which is then read by a signaling pathway. And that signaling pathway, that biochemical pathway, ultimately leads to a change in ionic fluxes in the cell. And that change in ionic fluxes leads to a change in the electrical properties of the cell and the generation of an electrical signal. And it's that signal which is then read by higher order neurons and the brain. Does this make sense? Now, the light receptor molecule catches a photon of light and undergoes its conformational change in around 20 picoseconds. This is very, very, very fast. Now, the photoreceptor neuron triggers an electrical response in around 100 milliseconds. This is 1 billion times smaller. So if the length of this pointer represents, let's say, the 20 picoseconds, you would have to draw a line that goes many times around the world to get a sense of the difference in time between what it took rhodopsin to do its thing and what it took the cell to do its thing. And that difference in time is, in essence, nothing but a reflection of the fact that you have this very complex biochemical cascade that needs to translate what happens to rhodopsin to ultimately what happens to the cell. Now, it is the unraveling of this biochemical pathway that was the focus of our work for many, many years. And that's because many known mutants in human retinal disorders are due, in essence, to nothing but misfunction of components of this pathway. And I think the next slide illustrates one such example, which is one of the most prevalent and perhaps, most common and devastating human retinal diseases known as retinitis pigmentosa. So this is a normal eye. This is the back of the retina, and this is the eye of a patient with RP. And RP is characterized by the presence of these big pigment clumps. That's the name retinitis pigmentosa. And in essence, this reflects nothing but the destruction of the underlying photoreceptor neurons. And what happens to these people in the end is that they lose all night sight, vision, and they become, as the disease progresses, then defective in their ability to also see during the day vision, because the loss of rods ultimately impacts the ability of the cone cells to remain in an intact microenvironment. Now, we want to understand how is it that the function and misfunction of the components of this pathway leads to such devastating disorders, and it's thus using genetics that we set up to dissect this pathway. Now, we could go around, look for patients that have different kinds of eye disorders and diseases, but it wouldn't do us much good because we won't be able to screw around with their eyes. Nobody will go and say, yes, take my eye, do what you need to do to it, and then give it back to me when you finish. And so instead, we rely on model organisms. And the great advantage of fruit flies is the fact that you could do, as I highlighted a few minutes ago, very unique manipulations. Now, the eye of a fly is fundamentally different than the eye of a person. But the underlying logic by which rhodopsin converts a photon into an electrical response is very, very similar. Now, a fly eye is made out of 800 ommatidia, they're called, or unit eyes. Now, each unit eye is made out of 20 cells, And among those 20 cells are 8 photoreceptor neurons. Now, we have millions of photoreceptor neurons in our retina. A fly has 800 times 8. So it's a much smaller repertoire. Now, contrary to what cartoonists want you to believe, a fly does not see 800 of you, nor does it see 1/800. There is extraordinary optics that ultimately puts the image back in the brain. Now, when we began this work many, many years ago, we knew about the light receptor molecule rhodopsin, and we knew about the output, which is a change in ionic permeabilities. So we knew that activation of rhodopsin leads to a change in ion fluxes, and the change in ion fluxes ultimately leads to an electrical response. And so our studies in the genetic of phototransduction were aimed at ultimately deciphering what is this black box. What are the molecules and the components that ultimately translate this signal here into the electrical response. Now, we've used many strategies to take advantage of flies, but the most important one has been the use of fly genetics. And that's because what you could do with flies is you could take flies, you could feed them a very strong mutagen, and now look for mutants that have defects in eye structure, form, or function. And if you do that, in fact, you can get a very large repertoire of either morphological or functional mutants. And this slide, I think, shows an example of various fly mutants with defects in eye structure. So this is the eye. You see, forms a huge part of the head, by the way, in flies, of a normal fly. And normal flies have red eyes. And you can get mutants that have, for example, different color eyes. This one has white eyes. And this one here has brown eyes. Or you can get mutants that have small eyes. Or you can get mutants that have almost no eyes. Or you can get mutants like this one here that has eyes at the tip of a leg growing out of an antenna, which is in the head of a fly. Now, creating these Frankensteinian organisms, it's not simply a source of great joy for us scientists, but more importantly, it's a strategy that allows us to ask what happens to this pathway, or what happens to this biological process when I now alter individual components. And by understanding what goes wrong when something is missing, we can then decipher how things normally work. Make sense? So you could take the steering wheel from your car and now realize that you can no longer turn. And so you could infer, the steering wheel must be important for turning. Now, you have to be wise enough to make the right inference, yes? It's like the story of the frog, where, you take a frog and then you pound on the table and the frog jumps, and then you take one leg out and then you pound and the frog jumps, and you take the second leg and you pound and the frog jumps. You take the third, you take the fourth, and it no longer jumps. And then you say, legs are important for hearing. Now, it's a long story, and I simplified a bit, but it's critically important that you decipher the logic of your mutants in the right context. Now, of course, we're interested not in eye morphology, but eye function, and so we use slightly different strategies. And what we do is to look for mutants that have defects in eye function. Now, there are many ways you could do this, but one of the simplest is by behavior. Flies like light. So you could take flies, you can mutagenize them, you feed them some of the sandwiches we had outside, for example, and then you now throw them towards the light. And flies like to go towards the light because they're positively phototactic. And you look for flies now that no longer go to the light. Now, among those flies, there'll be flies that have no eyes. There'll be flies that have no wings. They cannot go towards the light. There'll be stupid flies, flies that don't realize they should go to the light. But there'll also be flies that have normal-looking eyes, but their signaling pathway, this black box, is defective. And so they can no longer convert the energy of a photon into activation of the cell. Now, of course, doing that experiment is tedious because then you have to run around the room looking for the flies. And so we use slightly different strategy. And one of them is to use this device, which is I think everyone understands the logic of mutagenesis, which is known as a Y maze, where we can put flights at the beginning of a tube, and then we can put different type lights at the ends of the tube, and then even choose flies that can discriminate with specific types of light. Now, I also use this device to choose graduate students, because those that come to me with flies in the middle tube are the ones I do not want. Now, again, behavior is wonderful, but it's a little too far removed from physiology, which is what we want to understand. So we actually don't even do this behavioral test. We actually go to the heart of the matter, which is to stick electrodes in a fly eye and record light-induced electrical activity. This is no different than what you guys get when you go to an ophthalmologist to get an ERG, an electroretinogram. And so we developed means of actually not only recording from sticking electrodes in a fly eye, but actually from isolating the cells and being able to manipulate individual photoreceptor neurons by sticking electrodes in a way that we can really control their microenvironment. Now, I gear my whole talk at a very general level, but I need to give you a little bit of detail so the logic at least makes sense. So if you stick an electrode in a fly eye, and now you give a flash of light, you activate rhodopsin, 100 milliseconds later, all these biochemical events happen, and the cell depolarizes. The cell, in essence, lets ions in, and there is electrical responses. If you have the electrode connected to a very good amplifier, you can read that tiny amount of change in current that's flowing, or voltage, in that eye or individual photoreceptor neuron. And if you do that with a normal fly, which is shown in here, and you stick electrodes in the dark, you see no activity here, this flat line. You give a flash of light, and then you get a little spike here, and then you get an electrical response. You see this inward reflection represents the flow ions into the cells. And as long as you keep the lights on, these guys remain activated, and then you turn the light off here. You see this is the pulse of light. It goes on, stays on for 1 second, it comes back down off. And you can see that when you turn it off, they now repolarize back, they recover back to baseline. And this is what a normal electrical response to light would be in a photoreceptor neuron, in a fly, or in a human eye. Only that the polarity will be different in our case. All right? This is a collection of mutants isolated in genetic screens with selective defects in light responses. This you could see here is the most dramatic of all. You can give as much light as you want, and there is no response. And they're known as NORP, for no receptor potential. This one here, if you could see here, is known as a transient response. You see, you activate here light, they respond normally, you keep the light for a long time on, and these guys go back to baseline, even though the light is on, as if a component required to maintain the excited state has been exhausted. Make sense? And you have other ones like this one here, known as SLURP, for slow responses, where you turn the light on, they activate normally. Then you keep the light on, they remain normal. You turn the light off, and you could see they take a long, long time to recover. This is akin to you closing your eyes and seeing longer and longer and longer. And I mean not under the influence of any psychedelic agents, but actually as a result of the transduction process being unable to turn off. Now, looking at these mutants and then characterizing them physiologically and molecularly, getting at the individual defective component, allowed us over many years, in essence, to define this black box. And this work resulted, much to our delight, in the identification of nearly each and every one of the components required to convert the energy of a photon of light into the modulation of light receptor activity. There are altogether around 25 different genes encoding around 40 different proteins that are ultimately responsible for orchestrating the response of a photon of light in a photoreceptor neuron in a fly photoreceptor cell. All right? Now, knowing this, of course, now makes it very efficient and effective to begin now to study how misfunction of these components can lead to disease. And in particular, we can go and ask, how do defects in these components can either mimic or resemble, these are flies, remember, human either retinal or human neuronal disorders. And to make a long story short, let me tell you that it is the study of this type of pathway that's really shed significant light onto how this type of signaling cascades are ultimately regulated, not only in flies, but also in humans. Now, I'll give you one example of a unique type of disorder that is seen when you have signaling pathways like this one here that fails to be turned off effectively. Remember that slow recovery mutant? It turns out that activation of a pathway is as important as inactivation. Because when you have a hormonal response, you want to make sure, for example, that the hormone comes and then goes. When you take a drug that acts on specific receptors to trigger a pathway, you want to make sure that when the drug is gone, the response is no longer there. And there's a set of human disorders that come about because of defects in the way these pathways are being turned off. And I'm going to tell you about at least 3 or 4 states in which we could define precisely what are the critical components required for the inactivation. Now, examples in humans are a set of retinal disorders that comes about because you can no longer turn off your light response. There is this devastating disease known as familiar precocious puberty. These are little children which go through puberty at the age of 2, 3, and 4. And they go through puberty at such early age in their lives because specific pathways similar to this fail to either receive or to send signals that turn off appropriately. And then the one that you probably all heard about is various forms of hyperthyroidism, which again, your thyroid gland fails to turn off properly, and as a result of that continues to produce the T3 and T4 hormones that ultimately lead to hyperthyroidism. So by then simply looking at this incredibly unrelated pathway in flies has provided significant insight as to how each of those disorders comes about. And this is an example of those various steps I show you, where you can actually show that you can now go into these flies and engineer individual flies when a single one of these components is defective and ask what is the impact in the light response. And these are 4 different proteins in this complex cascade, each of which is essential for efficient inactivation. This is what a wild-type response looks like. You see it's very narrow, how long it remains open after you turn the light off. This is just a flash of light. And you can see this guy turns a little slow. This turns significantly slower. This much slower. And this extraordinarily slower. And it turns out that this molecule here is one of the most critical players in the overall phenomena of learning and memory. And so once again, understanding how these components work can begin to shed important insight not only on something like photoreceptor function, but the general signaling in neurons in normal and disease state. Now let me give you an example as to what happens when you have these guys defective actually in flies and in humans. So this is one of those components, this is one of the mutants that fails to be turned off. And this is an electron micrograph picture of a single unit eye of a fly, of the 800 units. In a normal fly, you see the group of 7 photoreceptor neurons, and this here is that organelle, that compartment that contains rhodopsin. And this is what a normal eye looks like. This is a mutant that fails to be turned off, but it's been kept intentionally in the dark during its entire life. So this fly has always been in the dark. And you could see that it looks no different than a normal fly. And this here, where everything now is being destroyed, is this very type of fly that now was let go into the light for 4 days. And after 4 days of exposure to light, you see the retina is completely degenerated. And that's because they activate normally, but they cannot turn off. And the failure of inactivation leads to an unusual metabolic imbalance that the cells ultimately degenerate. Now, we can now use this very same system also to find solutions to the disorder, either to alleviate it, cure it, or prevent it. And an example here is this very mutant fly in which we did gene therapy by introducing various forms of the normal gene. And you could see that there is a complete restoration of normal function. So let me then finish the photoreceptor part by simply illustrating that another great beauty of flies is that we can not only study this process in defective flies, but we can engineer flies where the same fly is part mutant and part normal. And the fly shown in here is a fly where the right side is female, red eye, and normal, and the entire left side is white eye, male, and mutant. This is known as a genetic mosaic. And so in the very same fly, we could have mutant and control and study the impact of the defects without having to worry about environmental consequences or any other artifactual effects. Now, to do this, all you need is tiny little scissors and a lot of patience. And so you cut the fly normal, you cut the mutant, and then you put them back together. You don't. You do this genetically, by the way. Okay? All right. So let me now move into Meccano Sensory transduction. So Meccano Sensory transduction, as the name implies, is a process whereby a change in the mechanical state of a sensor neuron leads to a change in the electrical properties of the sensor cell. Now, mechanosensory transduction underlies a wide range of senses, both in us and in flies. They are responsible for our sense of touch. They're responsible for our sense of hearing. They're responsible for our sense of balance. They're responsible for proprioception. Proprioception is your ability to tell, is the ability to tell the position of your limbs in space. I'm not looking at my hand and I know it's pointing up. And I know it's pointing up because I have sensors in all my joints that tell my brain my hand is pointing up. And the ability to do this is known as a proprioceptive sense. And then there is visceral senses. Yeah, I know that I'm full when I eat like a pig because my stomach wall has been extended, and then I have mechanical sensors that tell my brain my stomach is extended. Now, there is some amazing physiology about how mechanosensory cells function. And some of the best studies have been done in the vestibular and the cochlear organ, the sense of balance and the sense of hearing. And the man behind a lot of this work, this extraordinary physiology, is someone called Jim Hudspeth. Now, the cell responsible for detecting mechanical senses in our hearing and sense of balance is known as a hair cell. And it's known as a hair cell because it's a cell that has at the apical surface something that looks like a hair bundle. Now, these cells have extraordinary physiological features. And the basic, most important one is the fact that this little hair bundle acts as a mechanical sensor that upon deflection leads to activation of the underlying neuron. And so in the case of hearing, sound waves change the movement of fluid inside your ear, and that leads to the tweaking of this hair bundle. Now, these cells have three remarkable properties physiologically. Now, the first one is that the responses are extraordinarily fast. They take microseconds. So you can take one of these cells, you can tweak this little bundle, and within less than a microsecond, they trigger an electrical response. I told you just a few minutes ago that in the case of photoreceptors, it takes 100 milliseconds to trigger a response. And that's because you have this complex biochemical cascade. If it only takes a microsecond, there's no way in hell you can actually have a biochemical cascade. And this has led to a model, which is very important to know, which is a direct mechanical physical link between moving the bundle and opening a channel that lets ions flow and a change in the electrical properties of the cell. So first, the responses are very fast. Second is sensitivity. You can tweak this hair bundle, the one seen in this diagram in here, or in the slide before, a fraction of a nanometer. This is atomic resolution, and trigger a response. This is akin to you tilting the Eiffel Tower the thickness of your thumb. And the third unique property they have is they are directionally selective. If you tweak the bundle to one side, you activate the cell, and if you tweak it the other side, you inactivate the cell. Now, these properties have led to a model known as the gate on a spring to explain how you convert the physical tweaking of the bundle into an opening of ionic conductances. And this idea is very simple and is diagrammed in this cartoon. Here you have the hair bundle. Let's say this is the positive direction, so that's the way you're going to open the channels. The notion is that these tiny little protrusions going out contain ion channels that will let ions in, and in doing so will activate the cell. Let's assume that those ion channels are normally closed and they have a little gate connected to a spring. So when you have one of these little hairs sticking out next to the other, and you have a gate on one connected by a little link, a spring, to the other one, then you have them at rest. And now when I move them in the positive direction, you're going to have shearing between my two fingers, and in doing so you're going to stretch the little spring that connects them together, and in doing so you're going to open the gate. Does this make sense? And this opening of the gate happens now instantaneously. Lets ions flow in, and in doing so trigger a response of the cell. And this is illustrated in this cartoon here, where you have tweaking of the cell, you have the little spring, you have the channel, you pull them to the side, ions flow in, the cell depolarizes, it triggers an action potential. Now, this is a bit more physiological of an explanation, which illustrates the same idea, where here you have the guys at rest. This is the little ion channel. You tweak them to the right. That's the positive orientation in here. And you could see in doing so, this little gate here, which was closed there, it's now open. Now, conversely, if you tweak them on the negative orientation, you're going to really push the gate even further close. You're going to shut them even more. And in doing so, you're going to close whatever few channels you had at rest. That's why in one direction you go activation, and the other direction you go inactivation. Now, despite the amazing physiology done in the biology of mechanosensory transduction, very little was known about the underlying molecules. And the holy grail of the field was this ion channel, the mechanically gated channel. Because identifying the channel will give us a molecular tool to understand how this process once again functions in normal and disease states. And there is a wide range of human hearing disorders that come about because of defects in the function of this channel. In fact, it's generally believed that a lot of the damage that you suffer, hearing loss, through very loud noise is partly due to disconnect between the channel and the little spring. Now, so we figure perhaps we can help identify this channel using the very same logic, which is using a system suitable to genetic manipulation. And so we turn again to flies, and flies, it turns out, are very rich in mechanosensory organs. They have mechanoreceptors lining almost entire body. And that's because they have little bristles stuck on their body that sense wind direction as they fly. And by doing so, they're able to orient themselves quite effectively during flight and during landing and takeoff. Now, the structure of a fly mechanosensory neuron is very different in morphology to what happens to a vertebrate hair cell, but it's very similar in logic. You have an underlying neuron that contains mechanically activatable structure, where upon movement of the bristle, so this is one of the fly hairs that sticks up and goes all the way to the second floor here. And the notion is upon tweaking of this bristle, you impinge at the tip of this neuron, and this impinging creates a compression. Compression is read as a mechanical depression, and that mechanical depression is read ultimately as a mechanosensory stimulation that leads to activation of the underlying neuron. All right? So we wanted to know how close a model are flies' bristles to hair cells, so that if we can understand how this operates, we can ultimately be able to use one to understand the other. And so we develop a way where we can take fly bristles and record electrical activity from them. And so we came up with strategy where we can go into a bristle, we can cut the bristle, stick electrodes through the bristle into the underlying neurons, and then ask what are the physiological properties of those neurons. And much to our surprise and delight, we find that they're absolutely indistinguishable from a vertebrate hair cell, or to be more specific, from a human hair cell. They are directionally selective. So you take this bristle and you move it one way, you activate. You move it the other way, you inactivate. And if you move it on the orthogonal plane, you see absolutely no activity. And very much like you see in the vertebrates, they have extraordinary speed, so microsecond scale, and the same sensitivity, nanometer level. And so for all practical purposes, you can actually take this physiology from a fly bristle and superimpose them on the physiology of a vertebrate hair cell. So armed with this information, then we set up to look for fly mutants defective in Meccano Sensory transduction. The hope being that perhaps some of those are going to be defective because we mutated the channel, this holy grail of the field. And if we can find a mutant defective in the channel, we can then characterize a mutant, and in doing so, identify the channel. Make sense? Now, we needed a screen, of course. How do you look for mechanically defective mutants? And so we tried many things. Like, for example, can we take larvae as opposed to flies and have them walk on sandpaper? And they're going to feel the difference between normal surface and this rough surface. And larvae do not like that. So can we find larvae that don't mind it anymore? And they don't mind it because they're unable to sense the difference. Make sense? But it was too difficult to actually work it out in an industrial kind of way. So together with an extraordinarily talented postdoc, we came up with a different idea. And the fact is that if you take larvae, so larvae is one of the stages of fly development, yeah? And we didn't want to look at adults because we were worried that an adult, which is mechano-insensitive, might be so screwed up that we might never be able to get that fly out of its pupal case. And I'll show you that that's in fact the case. We have to get them out by C-section. So if you take a larvae, which is nicely wandering around, and you take an eyebrow and you hit the larvae on the head, the larvae sense that they're being touched, and of course they don't like it. It's like hitting you with a telephone pole in the head. And they have a very stereotypic response. You touch them on the head, and then they will withdraw their anterior. They have a single reverse contractile wave, and then they move away. And so we ask, can we mutagenize flies, get these larvae, and identify mutants now which deviate from this response? And the answer was yes. We could get larvae that go all the way from the energized rabbit phenotype, that it doesn't matter how hard we hit them, they just keep on going, to larvae that you touch once and they're completely paralyzed, as if they've been hyperactivated into a state that they cannot get out. And so we took this large repertoire of mutants, which was actually a significant number, and now we go and we ask, what tissue is defective for the larvae to be defective? Because we care about the larvae in which the peripheral sensors are defective, not where the brain is unable to read the sensors. And so by doing sophisticated genetics, we could identify all of those that have the defects at the periphery, and we can now begin to study among those which have physiology consistent with being a defect in the mechanically gated channel. Now, the next slide, I think, shows a picture of Morris, the person that did the work, which was one of the casualties of the work, I would say. I think before and after doing the study. And as you could see, he completely ran out of eyebrows because we, of course, use eyebrows as the means of stimulating the individual larvae. Now, there were many, many mutants, but there was one that was particularly intriguing because we had different states of the mutation. This is a normal response, so you take a fly bristle, you tweak it, and then within a few microseconds they activate. In this mutant, there is a complete loss of activation, and this is a different version of the very same gene. You could see that they activate normally, but they fail to remain active. And so through a set of physiological studies, we reasoned that this could be a component of, or actually be, the mechanically gated channel. And then when we go on and then we characterize this molecularly, lo and behold, much to our delight, this indeed turned out to be the mechanically gated ion channel. And so out of this screen in fly larvae came the critical component of mechanosensory transduction, which is now, in essence, the founding father of a family of mechanically gated channels, not only in flies, but also in vertebrates, including humans. So they are related members. Now, we have a few seconds, so let me show you a movie as to what these flies look like. And then if we have a few minutes, we can talk about taste. Okay, so here what we did is we put flies on a little dish, and we have a glass cover slip on top, so the flies don't escape. And then we have a microscope on top, so we're looking at the flies through this little glass cover slip, all right? And these are normal flies, and they do the kinds of things. Can we get the lights down? And they do the things that flies stuck there, as you could see. They do the kinds of things that flies normally do. They're happy little flies moving around. And you'll see a fly here that, they can groom perfectly well. They're grooming there with 2 legs. And you see a fly here, that one I think is grooming with 2 legs and then with 3 legs. So they have the right level of coordinated happy behavior that happy flies normally have. Now, this is the least effective of our mutants. And the first thing that we noted with these flies is that they turn out to be sedentary. So you saw the flies before, they're moving all the time, all around. And these flies, they tend to stay in their place. They don't really move much. And you'll notice that they tend to stick their middle leg out, but they can groom themselves perfectly well. And you'll see they'll tend to extend their middle legs out. If you focus on them, you'll see it. But they have another phenotype, which is the most remarkable. And I'll come back in a few seconds. So normally flies, will be just jumping all over the place. You'll never see them resting like this. These are all single mutations in single genes. Now, the incredible thing about these flies is that they cannot walk upside down. You see, they go up, the cover slip, and they this just kills me every time I see it. And you can see they fall flat on their back. And that's because the type of coordinated movement that you need, we changed the magnification there. You could see that one again. And so what happens is the type of coordinated movement that you need to walk upside down against gravity is very different than with gravity. And so the slight uncoordination of these flies is due to a defect in the mechanical senses. This is another mutant. This is similar to the mutation in the mechanically gated ion channel. And you could see these flies have a defect in a single gene out of the 16,000 genes in the fly genome. And this one gene leads to the complete paralysis of any and every coordinated movement that a fly can possibly have. And I think we're going to change the magnification in a second, and you're going to see these flies. And these are the ones which now, if you look at the other senses, they're perfectly normal. So they can see perfectly well, they can smell, we believe, reasonably well based on antennograms. But they are unable to perform any coordinated movement. And this is because they have a loss on the critical component for mechanosensation. And it's the same component that's required all the way from hearing to their sense of proprioception and to the sense of balance. And then there is one last. Now, you might wonder how you can propagate these flies, how you keep them going, because they don't mate very well, as you could imagine. So how do you do it? It's not like you can take one fly, you take the other fly, say, okay, mate, let's do it now. It doesn't work that way. So these flies are homozygous mutant. Flies, like people, are diploids. They have 2 copies of every gene. So to show the defect, then you have to have mutations in both chromosomes. So we keep them as heterozygous. We keep one good copy with one bad copy. And so in those conditions, the flies are perfectly happy. And when you cross them together, 1/4 of the progeny will be bad with bad. Now this now is one that's in between. And you can see these flies manage to get up. We changed the magnification there. Sorry about the resolution. This is one of the movies I lost as I changed computers. And so I had to put a lower. And oh gosh, they're just barely able to get up. But you'll see something quite extraordinary that happens with one of these flies. So you could say their sense of balance is slightly off. Now we're going to follow this fly that's going to get to the edge of the vial. All right? And you'll see in a second. And it gets, I think, right there to the edge of the vial, and it's running against the vial, but it doesn't get feedback saying you reached the end of the vial, and it keeps just pushing along, trying to go through, because it has no mechanical senses to say, I'm hitting my head, this is the end of the vial, let's move on with life. All right, so I don't think I have to show you the larval responses. You know, that's the larvae, and there's going to be an eyebrow from Morris somewhere there. There you could see the eyebrow. Can you see it? And you touch it, and it has a nice contractile wave, and they move away. All right. And so, once again, I hope that I have illustrated in a simple, yet perhaps reasonably clear way, how you can use straight genetics as a means of dissecting complex pathways, and in doing so, identify the critical players that mediate these fundamental biological processes. So in the last 10 minutes, let's talk about a bit the biology of taste. Okay? The sense of taste is responsible for detecting and discriminating among various chemosensory stimuli. In particular, sweet, sour, bitter, salty, and umami. Umami is the taste of MSG, monosodium glutamate, and it's what you often associate with headaches in Chinese restaurants. Let me tell you that there is no link between headaches, MSG, and Chinese restaurants. And so if you do have that problem, I recommend that you go to a better restaurant. Now, the unit of taste is a taste receptor cell. And taste receptor cells are located in your tongue epithelia in little clusters known as taste buds. And each taste bud contains around 100 to 200 individual taste receptor cells. Now, there is the general belief that there is a tongue map, and that different parts of your tongue are specialized for different tastes. And in fact, there are many companies that sell these wonderful wine glasses, which I may admit I love, which claim they maximize the way your tongue, given its distribution of receptors, can extract the most out of wine. It's utter complete nonsense. It's all olfaction. The wines do a wonderful job at concentrating the aroma, because we have now shown that you don't really have a tongue map. What you have is that different parts of the tongue have a slightly higher representation of certain taste qualities. But all areas of the tongue can respond and detect all taste modalities. All right? And so this illustrates what you see. So for example, bitter is really significantly more sensitive in the back, but you still have plenty of it everywhere else, and so forth. So our goal, when we began this work around 6 years ago, was to understand who are the receptors, who are the molecules in your tongue that are responsible for detecting sweet, sour, bitter, salty, umami, and translated that into a signal, again, an electrical signal, that these cells in the tongue can now send to the brain. And then ultimately what we want to be able to understand is, how does the brain know what the tongue just tasted? I know, and I'll show you how, the tongue knows. The tongue knows because it has cells that have receptors, and the taste and interacts with the receptor, and the receptor gets activated, and it activates the cell, and the cell sends an electrical signal. So that's how the tongue knows. But how does the brain know? It's not like you have a little guy inside your brain that says, oh, you activated the sweet cell. And so the logic of how that coding and decoding is actually the focus of our laboratory now, and it's been for the last 3-4 years. So our identification of mammalian, so now I'm going to switch to mice. And mice are very, 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 very good model for humans, because if you look at the gene diversity, there is really very, very little. And so our discovery first of sweet and umami receptors rely on two basic assumptions. The first one is the fact that the receptors has to be expressed in the tongue, because they're desired for taste, which is in the tongue. And so by looking for genes which are selectively expressed in the tongue versus other parts of the body, we figure we perhaps can identify tongue selective players. Make sense? The second was the assumption that a mutant defective in this one receptor will have problems tasting sweet. And it turns out that there were naturally occurring mouse strains that didn't like sweet. And naturally occurring means that someone went in the wild, found some mice 80 years ago, they bred them in the laboratory, and when they gave these mice a choice between water and water with sugar, where a normal mice will always drink water with sugar 20 times over water alone, these mice drank both equally well, meaning it couldn't discriminate between water alone and water with sugar. And so we reasoned that because the defect was only in that one taste quality and nothing else, that perhaps this is a mutation, a naturally occurring mutation in a gene involved in sweet reception. Make sense? And through a combination of this molecular strategy and genetic strategy, we identify a small family of 3 genes, which are known as T1R, T1R2 for taste receptor 1, taste receptor 2, and taste receptor 3, which, at least face value, were then implicated in sweet taste. Now, what we have here is an orphan receptor, a receptor whose function we don't really know. And so in order to know what it does, we need to develop a way to ask, does this receptor really recognize sugars? Make sense? But then we have a platform, then we need a platform to do that assay. And so we develop a strategy where we can take cells, normal cells, and we engineer them to change color when they're activated by just mucking around with their normal signaling pathways. And now we take these cells and we put these candidate receptors on them. And now we add tastans. And we ask, do they change color? And if we were able to match a receptor with its ligand, its tastan, then we hope that the cell will report that activity. Does this make sense? And so we developed such strategy. And much to our delight, we were able to demonstrate that out of these 3 receptors, 2 of them came together to build a broadly tuned sweet taste receptor. So 2 of these 3 genes made 2 polypeptides that assembled together and made a receptor that detected all kinds of sweet, both natural sweets like sucrose as well as artificial sweeteners like saccharin. Now, there were 3 genes, and so 2 of them came together to do this. It turns out that one of them could also function on their own to respond selectively to only high concentrations of natural sugars. So we have 2 types of sweet receptors in our tongue. We have one that responds to all concentrations of natural sugars and artificial sweeteners, and we have one that only responds to very high concentrations of natural sugars. Now, these receptors, by the way, are horrible receptors. It means that you need a lot of sugar to activate them. And they're meant to be horrible, because the role of the sweet detecting system in our tongues is to be able to detect high caloric food sources. And to do that efficiently, you want a receptor which is very poor. Because if you're trying to choose between 2 sources of sugar, and you have a receptor that's really, really good, then this receptor will very quickly saturate with either source. But if you have a receptor that's really, really bad, then it'll be able to detect the difference between these 2 sources, and in doing so, always ensure that you go for high caloric intake. Make sense? So when you put, therefore, 7 spoons of sugar in your coffee, it's not because you're screwed up. It's because your receptor is meant to be a very ineffective receptor. Now, this is a range of different sweets that we try, and every sweet that we have tried activates this one receptor. So out of the 3 receptors, then this combination, 2 and 3, defines sweet. But what about the missing receptor, which is 1 and 3? And so we reasoned, in addition, it turns out that in addition to sweets, there are some other fundamental components of our diet that might make sense to link to an attractive modality. So sweet is an attractive modality. We might or might not like sweets, but every other animal in the universe likes sweet. Sweet means attraction. Yeah? This is a bit like sex. We're only one of two species in the entire animal kingdom that have sex for the fun of it. Every other species has only one sole purpose, yeah? Which is to pass your genes to the next generation. So sweet and bitter are a bit like that. Every known animal in the kingdom, with the exception of humans, are averse to bitter substances. We, on the other hand, learn to enjoy them. Quinine, coffee, and the list goes on and on and on. And we might come back to that in 5 minutes if we have time. So what about 1 plus 3? So it turns out that we figure there's another fundamental element in your diet that might make sense to link to attractive modalities, and that is amino acids. Why? Because amino acids are essential components of our diet. We can only make a small subset of the 20 naturally occurring amino acids. And MSG, may I add, is an amino acid. It's monosodium glutamate. And so we wonder, is it possible that then this receptor represents the umami/amino acid receptor? And indeed, much to our delight, we found that if we take unrelated receptor and we challenge it with a whole range of amino acids, nothing happens. But if we take this receptor and now we challenge it with any of the 20 naturally occurring L amino acids, it all activated quite effectively. And so through these studies, we were able to demonstrate that the 2 plus 3 receptor combination makes a broadly tuned sweet sensor, and the 1 plus 3 combination is the mammalian amino acid/MSG receptor. Now, if this is true, this is all being done in vitro, in cells, in culture, then there are some clear predictions. And the simplest of all predictions is that a mouse defective in these receptors should be screwed up in its ability to sense, detect, and respond to either sweet or amino acids. Agree? And so we take mice, and now we engineer mice in which we ablate these genes. We genetically mutate the genes. And we ask what happens. Now, of course, you cannot ask a mouse, can you taste now or not? And so you could do one of two things. You could do the test I told you before, where you give them a choice between water and water plus sugar or water plus amino acid and ask what happens. Or in our case, we use a slightly different assay, which is a much more efficient assay, which is to take mice, you put them in a cage, you have them really thirsty so they're eager to drink, and then you present them with a repertoire, a collection of many, many different solutions in an automatic carrier that randomly switches between solutions. And then you simply ask, how many licks does the mouse do to any one solution in a fixed window of time? Okay? And the lick rate is a very good reflection of what the mice like. And in fact, this is what the apparatus in this case, looks like. And so there is the rack with all the tubes. And then it shifts randomly. And then there is a mouse in here that you barely see. And there's a little port that automatically opens. And when it opens, then the mouse has a certain window of time to lick. And then a little sensor detects how many times the tongue touch the spout. And then we just count that. And that is a reflection of the behavior of the animal. The nice thing about this is very fast. And so you don't have to worry about the state of mood of the animal. Is he happy? Is he unhappy? Did he have a good night? Did he fight with the other mouse and so forth? Now, so we do this, and now we knock out the receptor number 3. And receptor number 3 was a partner of the umami and the sweet receptor. So a prediction would be if you lose this receptor, you will be defective in your ability to test both amino acids and sweet. And indeed, here are responses of a wild-type mouse. So in this case, we have saccharin. And you could see that the more saccharin you give, up to around here, the more they leak. This is a normal mouse. Notice when you put a lot of saccharin, they begin to now dislike it. This is what happens to us also. It turns out that at very high concentrations of saccharin, you begin to activate a bitter taste receptor. And so the aftertaste of saccharin is due to the recruitment of a bitter sensor. All right? And that's why a lot of people don't like saccharin. And if you could see now the mutant mouse, it's a normal mouse, except that it's lacking that one gene, they're completely unable to detect the presence of the artificial sweetener in their assay. And this here is now the same for now amino acids, and then you see a wild-type mice, the more you put, the more they drink. If you put the mutant mice, they're incapable of discriminating amino acids from water alone. And this kind of data validates in vivo this one receptor 3 as the critical component of sweet and umami taste. Make sense? Now then we knocked out 1, and 1 is only a component of the amino acid receptor. And so you expect that you will screw up amino acid taste in these mice, but sweet should remain normal. And in fact, that's precisely what you get. These are responses to amino acids in a wild type, and you see the mutant is completely inactive. And these are the responses to sweet. And you could see wild type and mutant are indistinguishable from each other. And then finally, there is 2, and 2 is a component of only sweet, not of amino acid. And the prediction will be here that you will screw up the ability to taste sweet, but not amino acids. And that's precisely what happened. This is wild type, and you see the mutant no longer tastes sweet, but they can taste amino acids. So this type of experiments now validate, thus, the T1 nerve family of receptors as the sensor for amino acid and sweet taste in the mammalian taste system. Now, I'll finish with one quick slide. It turns out that mice and humans have some unique differences in their taste preferences. For example, mice do not taste a whole range of artificial sweeteners. And perhaps the best known among those is aspartame. So mice that do not like Diet Coke. Now, we reason that perhaps mice and humans differ in their ability to detect certain sweets because differences in the receptors. They live in different ecological niches, and perhaps changes in the receptors between mice and human could be responsible for taste preference differences between the species. And by extension, perhaps differences in our own preferences, the way my receptors differ from his receptors and from his receptors. And so we figure this could be testable. And the prediction was we should be able to clone the receptors from humans and now take the human receptors and put them into a mouse and ask, have we humanized the responses of the mouse? Does it make sense? And that's precisely what we did, and that's precisely what you get. And so we take this mouse that expresses now the human sweet receptor, and now this mouse has responses which are indistinguishable from ours. And so this, I think, if anything, not only is kind of cute, but more importantly, illustrate that differences in taste preferences are likely to be nothing but a reflection of differences in the amino acid sequences of the receptors among species. Make sense? Now, the bitter world is very different than the sweet world. Sweet, fundamentally, was designed to accommodate the need to react and recognize a single type of molecule, glucose and glucose derivatives. That's it. The fact that the same receptor recognizes a wide range of artificial sweeteners is of no consequence, because in nature you don't find them. Bitter, on the other hand, and that's the reason why you only have 1 or 2 receptors for sweet, because you only need to accommodate the need to recognize a very small repertoire of molecular identities. Bitter is fundamentally different. There are thousands of different molecules which taste bitter, naturally occurring, which have dramatic differences in structure. And in fact, as an extraordinary example of human evolution, and animal evolution for that matter, almost every known naturally occurring plant toxin tastes bitter to humans and is aversive to animals. And this makes perfect sense, because if not, you eat it and you die. And so there's a wonderful correlation between the bitter universe and nauseous tastes. And so here we reason that the family of receptors is not likely to be 1 or 2, but it should be very large to accommodate such a large repertoire of bitter taste. And here we rely not on mouse genetics or flies, but on human genetics. It turns out that you might have done this when you were little in school, where they give you this little substance known as PTC that they put in some sort of a paper, and they ask you to taste. And you can separate the human population into 3 groups. They are those that find it intensely bitter. They're called super tasters. There are those that find it pretty bitter. We're called tasters. And those which they cannot taste it, which are known as non-tasters. In fact, I fall into the non-tasting category. So when I taste this, it tastes like paper. There's nothing in there. And so Danielle Reed and co-workers a few years ago had mapped the human loci that segregated with the ability to either taste or not taste this compound. And we reason, given the specificity only for this compound, that perhaps this is a gene, a receptor, a bitter receptor for that one tastant. And so one of my graduate students, a terrifically driven student, Ken Mueller, set on to go into that human chromosome interval and ask, is there anything in there that could be a taste receptor? And to make a long story short, he was able to, in fact, not find one, but he found 3,000 related receptors, which accommodates the logic that you need to have a large family. And when we mapped them to either human or mouse chromosomes, every single one of them mapped to a location that in humans and mice had been implicated in defects in bitter taste. It's an extraordinary, revelation at the time we did the experiment because it gave you the answer right then. Now, if these are, of course, are bitter receptors, there is a predictions that we talked about before, which is that you respond to bitter tastants. And so we put them into the same setup where the cells light up if you find the right match. And in fact, we were able to identify receptors for specific ligands. And now, the next prediction is if we now go to mice and we knock out these receptors, either individually or all together, we should screw up the ability to taste either selective or the entire bitter universe. And that's precisely what we did. So we knock out one of the receptors that is specific for this one toxin, known as cycloheximide. Animals normally don't like it. And when we knock out the receptor, so a normal mouse, these graphs go down because the mouse are averse to the solution. When you knock it out now, the mice are unable to distinguish it from water. So this really shows that these receptors are indeed necessary for bitter taste. And then we can do the same experiment that we did before with with aspartame. It turns out that there are certain bitters that we can taste that mice do not. So we took some of the receptors for those bitters that humans can sense, we put them into mice, and now we can show that the mice can selectively recognize this repertoire of new tastants. This is an aspirin derivative. Aspirins have this very bitter component on them. And we can taste that, but mice cannot. So we took the bitter receptor gene that recognizes that, put it into mouse. So this is a normal mouse. It's indifferent to it. But now the engineered mouse is very strongly averse to it. And so these kinds of data demonstrates that these receptors indeed in vivo are both necessary and sufficient for bitter taste. Now, so then the repertoire of taste in humans then is that you have a couple of receptors for sweet, one for umami, and 3 or so dozens for bitter taste. And this is what we have in our tongue, and it's responsible for the logic of attractive and aversive taste. Let me just do one last experiment, which I think illustrates a rather remarkable logic of how taste is encoded. Not only by the way we remove individual bitter receptors, but we can also eliminate the entire population of cells, either by ablating them, by killing them, or by not allowing them to function. And if you do that for bitter cells, you find that you eliminate all bitter taste, but you leave sweet and umami intact. This kind of data is the one that proves that there is complete segregation between the modalities, because you can screw one up without affecting the other ones at all. And we can do the same experiments with sweet, umami, and bitter and show that they're completely independent of each other and of salt and sour. All right? Now, the next slide, I think, shows the importance of a bitter taste system. This is a wild-type mice, and wild-type mice are very strongly averse to all these bitters. These are all very bad news toxins. And now we took a mouse, in which we eliminated all sense of bitter by preventing the cells from signaling. And now we put them on a cage with these chemicals. And when we came back the next morning, that was the mouse. And so the loss of bitter taste, you could see, has an extraordinarily important role in survival. Because if you don't have it, of course, you're unable to recognize these very toxic substances. Now, I'm going to finish with this last experiment here, because you will appreciate the logic. So I show you about sweet umami, which are attractive modalities, and about bitter taste. Now, sweet receptors are found in sweet cells. Bitter receptors are found in bitter cells. Now, the question that we wanted to address was, are the receptors important, or is it the cells which are important in mediating the modalities? Meaning, is it the nature of the receptor itself that allows a tastant to have a certain character? This is sweet because it recognizes a sweet receptor? Or is it sweet because it activates a sweet cell which happens to have a sweet receptor? Does this make sense? And so we did an experiment to ask what is the answer. And the experiment was based on the very simple logic of how we believe coding operates in the taste system. If we reason, it is the cells that matter, and it doesn't matter how you activate it, as long as you activate them, you're now going to trigger that percept, be it sweet or bitter. Then we should be able to take a completely novel receptor, unrelated to taste, a synthetic gene that we made in the laboratory for a synthetic ligand, a non-taste containing molecule, and now express that in taste receptor cells which are either sweet or bitter. If it's the nature of the receptors that matter, then the mice are going to be indifferent to this novel molecule. But if it's the activation of the cells that matter, now the mice are going to be either attracted or averse to this novel compound, depending in which cells we express it. Does that make sense? That's precisely what we did by using a modified type of receptor, which is not normally found in cells. And when we put now normal mice and we treat it with this tastant, you find that the mice are completely indifferent. But now if we put this receptor in bitter cells, and now we challenge them with this ligand for that tastant, we find that the animals are strongly averse. We now take the very same receptor and we put it in sweet cells instead, and much to our delight, now rather than being averse, now the animals are strongly attracted to this very tastant. Now, a prediction of these studies is that if I now take a bitter receptor and I put it in sweet cells, now the animals should be attracted to that bitter tastant. Agree? And that's precisely what we did, which is the last experiment. So this is now this bitter receptor in bitter cells, and the bitter receptor in bitter cells triggers strong aversion. And now we take that very receptor and we put it in sweet cells, and now what you have is attraction to this otherwise bitter molecule. And so this demonstrates that it's not the nature of the receptor or even the tastant that matters, but rather the type of cell that you activate in your tongue. So sugar is sweet not because it is sugar. Sugar is only sweet because it activates this unique population of cells. Does that make sense? And so the concept, teleologically speaking, of sweet and bitter compounds is nothing but a reflection of the type of cells they express. And let me stop here because otherwise I'm going to keep on going. And I hope that I have given you at least a brief flavor for the kinds of things that that can be done.
It
it should be possible to train an animal to avoid sweet.
Absolutely. You could do that.
Can you do that to a person and then find out whether it still tastes sweet, but they know that they're not supposed to do it? Or does it
Absolutely, yes, yes. And that's not a peripheral process, that's a central process, yeah? And you can do this assays known as conditioned taste aversion, where you link the taste of a certain molecule with something really bad for the animal. And the animal learns to associate that taste with something very bad. And thus you can force animals to no longer like things that otherwise would very much enjoy. It's like when I was a child and my father forced me to eat these horrible foods. I no longer like them. Yes?
From your work and what you've shown here, what can be said about the, I guess the nonlinearity of some sounds, in the sense that your ear can hear very soft sounds and very loud sounds, and it's famously nonlinear, but maybe taste or eyesight or something isn't. Is that in these peripheral sense, or is that something in the brain itself?
In the case of sound, it's actually a property of the hair cells themselves. They have the unique ability to detect not only specific frequencies according to their location on the cochlea, but in addition, they have these built-in amplifiers. And so the sensitivity is a slightly different story. Now, in the case of taste, for the most part, it's, I would say, a digital system, which is, in the case of bitters for sure, either you sense it or you don't sense it, but you don't really discriminate between them, if that was what you were trying to get at.
Well, sometimes, like you said, you put 7,
Oh, sorry, that's a totally different, yes. Your brain ultimately needs to deal with this complex. Let me give you an example. We have mice here that have this one receptor in sweet and the very same in bitter. And they respond differently whether it's in the sweet cells or the bitter cells. What happens if you engineer a mouse that has it at the same time in the sweet cells and the bitter cells? What would the mouse do? Is the question clear? Well, the mouse, just to give you the answer, is confused. And the mouse is neither attracted nor averse, because it's getting both inputs. And in essence, the brain says, well, there's neither attraction nor aversion. But we can, if you want to talk a little bit afterwards. Yes?
I work with children who have learning disabilities and Asperger's, and children will tell you that they can't turn off the sense of a touch. So, can you comment on that?
I am totally unqualified to do so. Nevertheless, I will. It's never stopped me before. Based on the little data I know, it is most likely to be a central event and not a peripheral event. So there are two ways you could visualize mechanistically how this comes about. Either your peripheral organs that sense touch are unable to turn off the stimulatory signal, and so you keep on getting a signal that says, I'm still, or the peripheral organ got activated and normally deactivated, but the integrators in your brain are the ones that fail to ultimately respond normally. A lot of the data argues that these are all central events. Now, how they come about, central, yeah, non-peripheral, yeah.
Did you expect to see a temperature dependence of the magnitude of the signal, since these are ionic currents?
Yes, but not in the way that you're thinking. And so the K10, the shift in fluxes as a result of the changes in temperature that we would experience are very minor. However, I didn't get to talk about a whole world of thermosensation. And it turns out that not as a result of affecting the flux directly, but as a result of having thermoreceptors that modulate the activity of these other sensory cells, you have a temperature effect. So there is a lot of anecdotal data arguing that thermosensors in your tongue modulate the way you perceive certain tastes because they modulate the way the taste receptor cells ultimately signal. This is anecdotal data. Nevertheless, it's consistent with the logic that you have thermosensors. But your point, by the way, is absolutely correct, that you would see that under the right conditions. And in fact, if you look at the original studies on action potential conductance, you could see this dramatic effect of temperature. Absolutely, yes.
Okay, two, so one more.
Yes.
What happens if you try and like reverse the aversion of the bitter tastiness by sweetening it?
You do. You don't reverse it. That's what we do every day.
Okay, so it's something bitter. What happens on the cellular level then?
No, ultimately, these bitter cells are signaling to your brain, and the sweet are signaling to your brain, and somewhere inside, up the pathway, there is a summator, a comparator, yeah? At part of the circuit that says, is there more good than bad, or is there more bad than good? And we know this works, because that's what pharmaceutical industry has been doing for years to mask the horrible taste of a lot of the antibiotics for children, which is you put a lot of sweetener because a spoonful of sugar makes the medicine go down.